So welcome to this web. Well, indeed, this is being recorded, so I forgot to mention it. So welcome to this webinar. My name is Mathieu Vinken. I'm the chair of the in vitro and in silico toxicology specialty section of EUROTOX. If you would be interested in our activities, which I really hope you do, so please have a look at the website of EUROTOX. You can find us in the section of About Us, so you can see what our activities are about. So unfortunately, this year, of course, we are still in post-COVID times, so that's why we decided not to have like an in-person kind of event. So instead, we have this series of webinars. We started up in January, so we are now at the 5th of a series of 6 webinars. And actually, as you probably know, we will be having our in-person meeting as part of the ICT meeting in Maastricht in the Netherlands from the 18th to the 21st of September, which is also a EUROTOX conference, by the way. So we will be sending some information about this out later on. By the way, you can also follow us on social media. And today, I'm very, very happy that we have a very renowned speaker, I would say. So Nynke Kramer, since 2021, Nynke Kramer is Associate Professor in Toxicology at the Toxicology Division of Agrotechnology and Food Sciences Group of Wageningen University in the Netherlands, where she teaches toxicology, toxicokinetics, and ecotoxicological risk assessment. So before that, she also was an assistant professor in toxicology at Utrecht University, also in the Netherlands. So her research focuses on enhancing the uptake of in vitro models in chemical safety assessment by developing models extrapolating toxic concentrations in vitro cell assays to toxic doses relevant to humans and animals. So Nynke published over 40 papers in top-notch toxicology journals, and she also received a couple of very prestigious awards. She is an internationally recognized toxicokinetics expert, and I'm really not exaggerating because this is also reflected in her involvement in major European research consortia in the area of toxicology and risk assessment. And by the way, this is also the topic of today's webinar, namely next generation physiologically based kinetic modeling. So before I will give the screen to Nynke, some housekeeping information. As you already noticed, this will be recorded. So for those that would like to listen again to the recording, or for those that unfortunately, are not able to attend, you will be able to watch and to look at the recording that will be uploaded to the EUROTOX website. Also, at the end of this webinar, you will be given the opportunity for asking some questions. So what I would suggest is that throughout the webinar, you just post your questions in the chat. I will be following up on this. And then at the very end, I will go through the questions. And of course, you also will have your chance to speak up at the end. So with this, I would like to welcome you again. Please enjoy the webinar, and I'll give the word to Nynke. Nynke, up to you.
Thank you so much, Mathieu. I will share my slides now. Just let me know if you can see them okay.
We can see them.
Perfect. So thanks all for making it to this webinar. It's one of the many that have been in this series, and it's really meant as a background and educational lecture on this case on physiologically based kinetic modeling, and at least their use in what we call next generation risk assessment. So like Mathieu said, I'm Dr. Nynke Kramer and I work at Wageningen University. He's been very he's put the bar high for me now, but I hope at least at the end of this lecture that physiologically based kinetic modeling is both more understanding to most of you, but also particularly you might get excited to work with it yourself. So with that, just to note that it follows up on the previous speakers in this In2Tox webinar, and I will be actually touching on a number of these assays and of these tools that have previously been talked about, so particularly adverse outcome pathways in the kidney and the liver models, in vitro models. And so this really naturally follows up on those talks. And you can look back at those talks on the EUROTOX website. So the presentation aim here is at least to answer the question, what are physiologically based kinetic models and how can we use them in next generation risk assessment? And in so doing, I'll touch on some of the opportunities, but also some of the challenges that we still have and still face. Of course, this might be quite gibberish for you, but let me just introduce them in a stepwise manner. So I'll go into why we even need to look at toxicokinetics or kinetics in toxicity testing in the 21st century and next generation risk assessment. Then I'll go into the basics of what constitutes a physiologically based kinetic model. And this is important to understand how we can apply them in risk assessment. I'll focus on quantitative in vitro and vivo extrapolation for a bit, just to illustrate how these PVK models can be used to do this type of extrapolation. And then I'll come up with some conclusions. And in the end, what should become clear is that there are many opportunities and also some challenges that we face. So as you will know, traditionally, risk assessment is done on animals, and we usually take a rodent, for example, and expose them to our chemical of interest, and we assume that humans are approximately, depending on the type of risk assessment, 10 times more sensitive than the rodent or the animal species, and that the sensitive human is 10 times more sensitive to the toxicity of a compound than the average human would be. And so this is how we derive, for example, a reference dose, like an advisable daily intake or tolerable daily intake. What this means is that we apply these uncertainty factors in reference dose settings. So generally speaking, we take the NOEL or the benchmark dose and we divide it by these factors. And these factors can be separated out into two aspects. So we know that there are differences in how you and me react to a compound, and this can be due to the kinetics of a compound, right? So what the body does to the chemical, and can also be due to differences in the dynamics of the chemical, so what the chemical actually does to the body. And these have been separated out in these uncertainty factors that you can see here. This is not necessarily scientifically based. We would actually want to understand what really are the differences between species and between you and me in kinetics and dynamics. So with kinetics, just to remember, because I will come back to that a lot, with kinetics, we mean what the body does to the chemical, and this includes the absorption of a compound, for example, through your gut, your skin, and through your lungs, the distribution, usually through your bloodstream, and then metabolism, usually in the liver, but also in the gut and some in the kidney, for example, and the excretion routes. And so all these aspects, you and I can differ, and we species can differ. And so the problem here really is it's the traditional way we do toxicity testing is a bit of a black box model, right? And this is particularly where we're actually, from a scientific point of view, we're not quite sure how to interpret it mechanistically and therefore assess how that it's relevant for us in humans. And this brings along with it, it costs a lot of animals, and we have a number of legislations here in Europe, at least, that actually where we're no longer allowed to use these animals. And so this is where toxicity testing in 21st century come in. And this is a landmark report in 2007 by the National Research Council. And really the basis of this report is that in vitro tests form the basis of toxicity testing in the future, because it's high throughput, little waste, you can use human tissue as opposed to animal tissue, and therefore it abides by the 3 R's, the reduction, replacement, and refinement of animal testing. And importantly, and this is what I would like to stress, it's a mechanistically based approach, which has already been alluded to by both Mathieu Vinke last time and Roos Meseret before, at least compared to the liver and the kidney. So these in vitro tests are making a move in regular toxicology. This is one of the very first studies that I did myself. I come from EUROTOX, from environmental chemistry, and I actually compared in vitro effect concentrations that we tested on for a whole suite of compounds to the in vivo, so the fish acute bioassay. And as you can see, there's a relatively good correlation here with a couple of outliers which can be explained by their specific toxicity, and we can accommodate for that. And actually, this first in vitro in vivo extrapolation here is actually now ingrained in an OECD guideline, which is the test guideline 249. It's been out since last summer. However, when we look at systemic toxicity endpoints and human toxicity endpoints, we have very few validated in vitro assays that we can use. And I think one good example of why this may be is the example of ToxCast, which was set up by the US EPA and the FDA and the NIH. And they've screened 10,000 compounds in a whole suite of in vitro assays using these high throughput robotic in vitro systems. This is the setup that you can see here. And the idea here is to screen for the potential of these chemicals to disrupt pathways, and therefore prioritize what chemicals to look for, to prioritize chemicals for risk assessment. But as you can look at this now slightly older study already, but it didn't change in that sense. As when you look at all these tested chemicals and you set that out against the concentration, the median effect concentration, activity concentration in these in vitro assays, it's very difficult to compare or prioritize the chemicals based on their supposed risk from these in vitro assays. So what's missing? So what we need, and this is where next generation risk assessment comes in, is we need more than just these in vitro assays. We need an exposure-led, hypothesis-driven approach to support these animal-free safety decision-making. And so in this case, we would use in vitro assays as well as these in silico or computational approaches. And this is where adverse outcome pathways come in, right? So, in vitro assays are great at understanding the interaction at the target, but how does that relate to an effect on an organism, or if you're interested in ecotoxicology, an effect on a population? And this adverse outcome pathways is really the framework to combine the knowledge that we have associating a molecular initiating event through effects on cells, on tissue, and the organism. So that's covered already in the previous talk by Penny Nijmark earlier. But what is still missing is that a toxic concentration in these cells, in these vitro assays, is still not really a toxic applied dose, right? A milligrams per kilogram body weight per day that we can take in that would be safe. And this is because we need to translate these concentrations into an external dose. And that translation is what where we need to understand toxicokinetics. So it's really understanding how we go from, for example, an oral dose to a concentration in plasma, to a concentration in tissue, for example, in the liver, and then ideally even to the concentration in the cell at the target, for example, at a receptor. In fact, this kinetics also needs to be applied in your in vitro assay that you're testing to, although this is not going to be the focus of this particular talk, but it is an important one to realize, because a lot of chemicals, the nominal concentration is not necessarily the concentration that's freely available in the system, and not the concentration that's actually found in the cells at the target. And so both in vitro and in vivo, we will need to take into account this, the kinetics. So, the extent to which a compound moves from an external environment into an internal environment. And this is where tools for these tools come in. So both for accounting in vitro kinetics, we can use in vitro distribution kinetic models, as well as in vivo kinetics, we can use physiologically based kinetic models. And so these are models that actually, compartmentalize the human body, and I'll go into that now. So this is PVK models are actually a series of mass balance differential equations that simulate the transport. Oh, sorry. That simulate the transport of a compound through a series of compartments that represent your different organs, right? And so they are parameterized using physiological model, physiological parameters like your blood flow rate, the volume of your tissue, the fraction of the blood that goes to the tissue, and these partition coefficients, how much does the chemical like sitting in your tissue as opposed to the blood. So like I said, so these are compartmental models. They can vary in structure depending on your interest and what you're trying to model. This is an example, for example, of a chemical that is inhaled through the lungs. So a fraction of the concentration that gets into the air in the lung goes into the blood of the lung, then the blood circulates around, and a fraction of that blood then goes to the different tissues in the body. And these can be specified as you can have hundreds of these tissue parts or tissue parts, but you can also have it very simplified and just separate out your excretion compartments from your non-excreting compartments, for example. When we look at the type of equations that go into these PVK models, it can look a bit daunting, but in fact, it's actually quite simple, and it's similar for every of these compartments, right? So the concentration in venous blood in the brain is equal to the concentration in the brain divided by this partition, this tissue blood partition coefficient, the extent to which a chemical likes sitting in tissue compared to blood. And so the concentration in the brain is a function of the amount in the brain divided by the volume. And so the amount in the brain is a function of the change in the amount of the brain, of the chemical in the brain. And this is then your differential equation, which is determined by your blood flow rate, if it's perfusion limited at least, and the concentration of the chemical in your arterial blood minus the concentration minus the blood that flows out of the brain in your venous blood, right, and the concentration in that blood. And so this is how these compartments are set out. So to summarize here, you have model parameters. They are separated into three categories, if you like. It can be organism properties and physiological properties, right? So these include your volumes, your tissue volumes, your blood flow rates, your cardiac output, etc. But they can also be chemical properties. And these tend to be properties like your plasma protein binding. So these are chemical specific, your tissue blood partition coefficients, and active processes like clearance of the chemical in your liver. And of course, there's the last category, you have these exposure properties, how often do you administer the chemical? Is it applied through the skin? Is it inhaled through the air, etc.? And you would put these into your models as well. A lot of these chemical specific parameters can be estimated using quantitative structure activity relationships, and I'll briefly go back into that later. But so chemical properties of your compound are very important here: the lipophilicity, the log P or D, in your case, the molecular weight, and the acid binding constant, acid association constant. So what you do in these PVK models, you normally put them in a numerical solver. Common ones are Berkeley, Madonna, R, MATLAB, for example, and it used to be Axel as well. You put your parameter estimates here, and then the equations in the differential equations per tissue. And what comes out, in fact, is here concentration time profiles in the different tissues. In this case, this is toluene in rats, for example, and toluene is absorbed through the lungs very quickly and then decreases in the blood very quickly because two things get metabolized, but also because it gets taken up into fat tissue. And what makes it so very neat, so instead of having just blood concentrations, you can really see how the concentration of your chemical and the patterns differ between the different tissues. And this will also allow you to identify target tissues, for example, because of a chemical accumulating in certain tissues, whereas they don't accumulate in others, for example. I've got an example of where PVK models can and are used, in fact, is for example, to set chemical specific adjustment factors. This is a very early study in the group that I'm here in Wageningen on coumarin, but it's a neat case study where, like instead of using that uncertainty factor of 4 to account for differences in species, the species differences in toxicokinetics, we can actually use these PVK models to actually tell you what the expected differences in sensitivity between the animal study and the human study. So, for example, coumarin, it's a hepatotoxicant, at least the metabolite here, the hydroxyphenyl acetaldehyde, is responsible for liver tumors in rodents, at least. And then the coumarin also gets metabolized through cytochrome P452A6 into 7-hydroxycoumarin, which is a detoxification mechanism. Now, what is interesting in rats and humans is that the dominant pathway in humans here is this formation of 7-hydroxycoumarin, whereas the dominant pathway in rats here, or at least a little more dominant, is this OHPA right here. And so it forms liver adducts and therefore these liver tumors. And so what you can do in next generation risk assessment here is you can use different liver tissue from different species, in this case humans and rats, and what was seen here when we did these microsomal these metabolite formation assays is that in humans we have much higher concentrations of this non-toxic metabolite, whereas in rats you have a very high concentrations of this toxic metabolite, which is not seen in these human microsomal chromatograms. And so when we input these values, these clearance values, these metabolite formation rates into a PVK model, we can simulate the concentration of the toxic metabolite in human, in the different tissues in the human, and we actually find that at least in plasma, the concentrations of this toxic metabolite is actually 1,600 times lower than in the rat at NOEL levels. And so you can also argue is there are people who are poor metabolizers of cytochrome P452A4, and so there's, but even when we model these individuals, they're still 20 times lower. They'll still have 20 times lower levels of the bioactive metabolite compared to the rat. So instead of using an uncertainty factor to account for the differences in kinetics of 4, we should be using an uncertainty factor of 1 in this case, or in this case we use 1 so that we said there is no difference here. And this also would mean that the tolerable daily intake of this compound is now not 0.4 but 0.1 milligram per kilogram body weight per day. Another aspect of PVK modeling, which is very valuable in risk assessment, is that we can make the assays, the models population based, and that's using a Bayesian approach. For example, there is a distribution in each of the parameters, so both a distribution in the physiological parameters and topical parameters like body weight and liver mass, etc., and also a distribution in your chemical specific parameters. And so you can use this as an input into the PVK model. And so instead of getting a single simulation of a concentration time profile, you would get the range at which you expect your concentration to be in over a population. And you can do this for different population groups, for example, and this has been very valuable indeed. And a lot of the research in this group focuses on this type of modeling. Now, for most of you, I can understand you're not going to make your own model, but there are quite a few generic PVK models out there. So these are models that have set a set of tissues that are compartments representing the tissues, and have different methods of estimating input parameters, for example. And I've listed common ones here. This is not, there are more than this here. In fact, the one that we use a lot here in Wageningen is the QIVIVE tools. .word.nl, but here are the most common ones. So HTTK is used by the US EPA, ARVIS is set up at the UK Health and Safety Laboratory, Plethum we have also as a part of Cytovation, and the SimSip simulator is not open source, but it's one of the most powerful generic models that have been used, particularly in the pharmaceutical industry, and the same is true for PKSim. Actually, so it's quite a lot of generic models that are out there, but like if you're looking for which one should I be using, so how do these models compare? And so there have been a couple of studies out there that compare these different generic models. For example, this one in pharmaceutics and biopharmaceutics just 2 years ago, and they looked at the predictive performance, and they found that the predictive performance, they actually didn't differ very much between the different generic models. It was actually the choice of input data that made that explained the differences here. So here you can see the fold error change in the area on the curve of these generic models compared to experimental data against a number of these chemicals. And in fact, in a very recent study that we did, and that is now currently in preparation for publication, we also did the same and looked at a couple of these generic models and found, for example, that again, the way the models predict, for example, the Cmax or the area on the curve compared to the dose was actually very similar. And what explained these big differences, for example, here, HTTK seemed to have a very different value for coumarin that I just mentioned as the compound. And that's because they assume that clearance is 0 liters an hour, as opposed to the other models. That makes a difference. As soon as if you change that parameter, it comes much closer. So that brings me to a challenge in next generation PVK modeling. So you have to imagine next generation PVK model actually is different from traditional PVK modeling in the sense that it solely relies on in vitro and in silico methods to parameterize and therefore also validate our PVK models, right? So traditionally what we do in PVK models is we calibrate and we evaluate the models based on in vivo data. So it's really a balance between the principles of parsimony, so in other words, the minimal but essential elements characterizing the system, and the plausibility, right? So the reflective physiology, it's a reflective of physiology, and it has to be consistent with the current stage of knowledge. And so here, when we talk about next generation PVK modeling, we're really talking about the models that are solely calibrated and evaluated on these in vitro and in silico methods. And so it really has to reflect a mechanistic understanding of the biology. And what comes with that is we're dealing with unfamiliar uncertainty, right? So uncertainty related to the relevance, the reliability, and the variability in vitro and in silico methods from which model parameters are generated. So next generation PVK model parameters. So how do we parameterize this, right? So I've put up a scheme here. It would be in a tiered approach. So a scheme here is like, what are the parameters that we need to estimate and be certain about, and or have an understanding of their uncertainty. And so these include the general absorption, distribution, metabolism, and excretion parameters here. So for example, impairment permeability, the tissue blood partition coefficient, the unbound fraction, the clearance, and generally speaking, in a tiered approach method, you have a general model here where we assume excretion is solely driven by the glomerular filtration rate, for example. A lot of these parameters are estimated, at least in the first tier, using quantitative structure-activity relationships. The most common one are tissue blood partition coefficients. So, for example, you can use Joost de Jong for more lipophilic semi-volatile compounds, for example, which does pretty well. But there are, unfortunately, to make it more confusing, quite a number of these quantitative structure-activity relationships, and their chemical applicability domain will differ slightly, and this is not very clearly reported. So that does make it more difficult to use and to assess for how appropriate they are. There are a couple of parameters that are very difficult to at least for now, difficult to use QSARs for. So these tend to be, and the most important one is actually clearance. It's in most of the studies that we see, it's one of the most sensitive parameters in these PVK models, and so in vitro metabolism assays, substrate depletion assays, and metabolite formation assays are used here. And what you would need to do, so that some of the assays that, for example, Mathieu Vinken in the previous lecture already referred to, are primary human hepatocytes, for example, and you'd have to then translate an intrinsic clearance in these in vitro assays to an in vivo intrinsic clearance, and that's generally, we apply scaling factors here. So, you know, the number of hepatocytes per gram of kidney, and then multiply that by the grams of liver. I've now here made an overview of the different in vitro assays that are being used or have been used to parameterize PVK models, and you can look, it's an older study, but it hasn't really changed much since then. But you can look at the KECO2 assay, which is generally has been used quite frequently to assess the oral absorption of a compound and you know, estimate this flux across the gut. Then there are a couple of dermal in vitro assays and inhalation assays, although I say that that's not been used much for now. Distribution is generally easier to parameterize, at least the information is more readily available. So like I said, partition coefficients is usually done using tissue partition coefficients is usually done using QSARs, but it can also be protein binding, and there are a number of standard assays available, like the rapid equilibrium dialysis assay or ultracentrifugation, for example. And for the more lipophilic compounds that we do in our lab, we actually use solid phase microextraction to estimate, to get good estimates of plasma protein binding, for example. Like I mentioned before, metabolism is generally parameterized using these human microsomes or human primary human hepatocytes. And there are also some examples of hepRG and hepG2. And excretion is generally parameterized based on just only glomerular filtration rate, but there are a few papers out there and models out there that will incorporate active secretion and reabsorption, which for quite a number of compounds could be is a dominant process in determining the kinetics of a compound. And this is actually one of the things that we look at in our lab now. So it's one of the challenges, I would say. So it's the active transport and its role in clearance through the kidney. Generally speaking, we would use these, there are a number of kidney cells, and these have also been explained in the previous lecture by Roos Massareo. There are a number of cell lines available, or tissue, but what we will find is like it's still very difficult to ascertain these scaling factors. So we have to go from, and we need what we call relative expression factors or relative activity factors. You know, we have to go from a clearance in vitro again to, you know, we have to understand the number of transporters per in the proximal tubuli, for example, and translate that to the transporters in vivo in the tubuli. And I've got a study that I put up here. That's a recent study that was done in our lab, where SOC7 cells were used to represent the the proximal tubule cells, and these have been transfected with the OCT2 transporter, which is responsible for the clearance of MIPIQUAT in both rats and humans. And the PVK model was established here to understand, and we incorporated these in vitro assays, the Vmax from these in vitro assay to ascertain this active secretion. And when we use a full scaling based on what I showed previously, based on what we understand to be, the amount of transporter per gram of proximal tubule cells, for example, we overestimate the concentration of MIPIQUAT. So we are still, in most cases, we have to rely on in vivo data to properly simulate the clearance of these chemicals in the body. So there's I've summarized it here. There's difficulty in ascertaining scaling factors, particularly milligrams per protein per a gram of kidney, for example. And we even less so, we don't understand the human variability in these parameters here. So just to sum up here for you now too, so the steps in that a tiered approach would take in next generation PVK modeling is we start with quantitative structure property relationships, and where not available, we use in vitro kinetic assays to be more certain of the parameter estimates that will go into PVK modeling. And then we still have the issues like, okay, but we now have our in vitro effect concentration. We still need to know what the effective dose is. And this is where quantitative in vitro and vivo extrapolation come in. So once we have this PVK model, we integrate that with the effect concentration that we find in vitro toxicity assays to ascertain the in vivo human toxicity estimate in this case. This is one of the main applications of PVK modeling in next generation risk assessment. So that's quantitative in vitro and vivo extrapolation. But I've also already mentioned here, this is to summarize. Currently, PVK models tend to be used to extrapolate or to do dose-dependent effects of kinetics, right? To understand species differences in kinetics and to derive these chemical-specific assessment factors, or to simulate what would happen using different exposure scenarios. That was related to that is like we can use these PVK models for root-to-root extrapolation, where we might have an understanding of the kinetics when we give a chemical orally, but we want to understand how it changes if we give it, if it's taken in through the lungs, for example. And often it's used to understand how patients will react compared to healthy adults, for example. And in a new, in the new setting, there's one good paper out by Alicia Paini where they actually apply PVK models in a read-across fashion. But in light of time, I can't go into that there. What we do, one of the most important applications is quantitative in vitro and vivo extrapolation. And just for your understanding, so this, what we mean here is different from in vitro and vivo extrapolation, just to confuse you. It's really the estimation of a chemical exposure producing a target tissue exposure in the species of interest, which is equivalent to the associated effect in vitro toxicity tests. So how do we go from a concentration response relationship to a dose response relationship in humans specifically, and how can we then use that as our point of departure for setting, for example, a reference dose? And this is a very neat example. It's one of the very first examples, and I still put it up because it's one of the neatest, I would say. It's done by Johan Louise in our lab in a while back now. And what he did is he looked at glycol ethers, and glycol ethers, at least their metabolite, the alkoxyacetic acids, are very embryotoxic. And so he tested these metabolites in a mouse embryonic stem cell test, to obtain these concentration response relationships. What he did, he set that equal to the concentration in fetal blood here. And then what he did is he using a PVK model in this case, he back extrapolated what the oral dose should be in the mother rat of the parent compound that would lead to concentrations of the metabolite that are toxic in this in vitro assay. And so the output here is a dose response relationship in the female, the mother rat. So, for example, in this case, the glycol ether, this is an actual animal study. These dots right here, and these are two forms of simulations of this median effect of this quantitative in vitro in vivo extrapolation study, and you can see it's spot on in this case. So, without using animals, you can, in theory, derive the same concentration dose response relationship as you would in an animal study. Of course, this is where it worked very well. That's why I like to show it. It illustrates it well. But remember ToxCast that I'd shown you earlier, right? Where they've done a beautiful set of beautiful data, but it was very hard to actually distinguish between the chemicals in terms of which chemical is a priority. That's because you have a whole range of concentrations what to take. But when they used this quantitative in vitro in vivo extrapolation approach, right, so they used a simplified PVK model here, they translated these effect concentrations to milligrams per kilogram body weight per day. And you can see here that there is a more clear understanding of the difference in expected toxicity range of these different compounds. And note these little green dots here. These are actually exposure estimates of the compound. And for example, you can see triclosan here is in this case a priority compound because the estimated exposure seems to be in the range that lead of the dose range in which we expose that could, at least in vitro, lead to adverse effects. So just in conclusion, so next generation PVK models rely solely on in vitro and silico methods for parameterization, but also therefore for validation, right? Because the idea is to use it in the future where we do not have the animal study data to validate the plausibility of the compound of the model. So PVK models translate in vitro effect concentrations to in vivo equivalent exposures. And this is what we call quantitative in vitro in vivo extrapolation. And PVK modeling allows us to characterize, and this makes it a very powerful tool, the uncertainty and variability in the sensitivity, at least due to toxicokinetics, as we've done in the past now, but also in the future for toxicodynamics. And in light of time, I can't go into that, but there is the integration of in vitro assay. We can use in vitro assays for variability, understanding the variability in toxicodynamics, and input that in PVK model to actually get a complete view and assess these chemical specific uncertainty factors. With that, I like to end this presentation. I just want you to note that there's a next webinar coming up, which is Tuesday, June 14th. And this will be Marcus Frerichs, who will give us a talk about QSARs and read across and how you can use them for risk assessments of plant protection products. You can sign up on the EUROTOX website, and so we hope to see many of you there. For now, I'd like to say thank you for listening, and I'm very much open to have as many questions as you might have. Thanks.
Thank you, Nynke. As always, excellent. Thank you so much. So we did receive 2 questions from one of the attendees in the chat. So let me first go through this. So the first question would be, what is the efficiency of PVK or PVK prediction in general?
So how efficient? Yeah, so
yeah,
that's it's a charged question, absolutely. So it will obviously depend on the model that you're looking at. But then I'm assuming here you're talking about on generic PVK models. And there, it's a study that I'd shown you briefly on there. So there's a general consensus, at least in pharmacology, that it has to be within a factor of 2. So for example, your AUC or your maximum concentration in a particular tissue of interest should be predicted within a factor of 2. In the toxicology community, we're also depending on, like if you're on what tier level you're going at, a factor 10 would also work. And there's a good study by Ans Punt that was published a few months ago, where she's actually compared, compared models and compared our models and for a whole suite of compounds to see how indeed how accurate these general predictions are in the first tier, and then they will fall. Many will fall under within a factor of 10.
Okay, thank you. And I think the second question from the same attendee is actually very similar. So the question is, and I will just read it out loud. So to what extent is the PBPK modeling prediction correlating with data gained from the wet lab experiments?
So I'm assuming here again, wet lab experiments are your animal experiments, or human, better yet, human clinical.
I'm not pretty sure. So if the attendee would like to speak up, that might help to clarify. If not, you can already answer the question in both scenarios, I would say. Nynke?
Yeah. So again, it's the same answer that I mentioned before, and what I'll just do now, I'll put in the paper in the chat. It's not the only one, mind you, but okay. Here they looked at the next generation PVK model predictions in rats based on in vitro and in silico data, and compared that to available rat in vivo data, and to see, like in what case, when how much fold difference is there in the Cmax. So based on these input data, right, they did 32 compounds, and they predicted within a 10-fold of the observed Cmax. So most of them actually were even within 5-fold or 2-fold, although there were some overestimations, more than 10-fold for 12 compounds, and it goes into the reasons for this. So there is still a way to go, of course. But if we can understand, and this is the next step, if we can understand the applicability domains, both from a biological applicability domains, right? When can we trust it being okay, and chemical applicability domain, we can add more trust in these systems. Okay,
thank you. And I invite again all people to post questions, or if not, to speak up, and maybe just awaiting other questions, a question from my side, a more practical question, Nynke. So as many of the attendees probably, my team also is focusing on elucidating mechanisms of actions of toxicants. And of course, we are always very keen to set up these kind of studies, and we know very well what we need to do from the dynamic point of view to have like the optimal setup and the optimal design in order to have a clear idea about the mechanism or the mode of action, if you will. So unfortunately, we tend to ignore the kinetic parts, which is again very, very unfortunate. But as we heard today, it seems to be like, an area on its own. So what would you recommend people like me and probably many other people, if you set up these kind of typical toxicity studies where you want to focus on, does this compound affect this kind of cell, and what would be the mode of action? So what would you recommend from your perspective to have at least the most sound kind of kinetic, let's say, setup? So in order to have like the results being to the best extent possible, or the most extent possible to be in vivo relevant. So what would you recommend? What would be the parameters that we need to focus on, or that we need to pay critical attention to?
Thanks. That's it's a really relevant question, and so I'm always an advocate of and I guess there are a couple of us. You're absolutely right. It's two separate worlds, not just in toxicology, also in pharmacology. It's but the modelers and the in vitro toxicologists and people looking at dynamics should interact already from the start. So a stepwise approach that I could now think of on the top of my head. The first would be, say, if you're interested in a compound or in an effect, you can use PVK modeling in a very rough way, right, to understand which target organs are likely, for example, because it will give you an indication where you can assume accumulation occurs, and that already gives you an indication where to look. And what it also does, it gives you a range of concentrations that are relevant for testing in your in vitro system. Right, and so if that associated at least with the exposure ranges that you have, and then the next and the second step is like, once tested, you would also go back and extrapolate the found effects to a dose that would be relevant for you to set, for example, a margin of safety.
Okay, thank you very much. These are very, I would say usable, recommendations that we can easily implement, I would say. So I think the take-home message would be, don't ignore the kinetics, which we, again, frequently, unfortunately, tend to do so. So I'm looking at the attendees. Are there any further questions? Don't be shy. You can speak up, so you don't necessarily have to include this in the chat. This is your chance. If not, then I can only conclude that it was very clear, and that way that there are no further questions. So with this, I think we can wrap up. And of course, I would like to thank you, Nynke, for another excellent webinar. I also would like to thank, of course, Elaine. She's always very, helpful with the setting up these kind of webinars, which is already the 5th one. And then, of course, I would like to thank the attendees for sticking with us and for asking questions. And if you would have any additional questions, then I can assume, Nynke, that I can directly contact you via email. Again, this presentation will be uploaded to the EUROTOX website, so you will have your chance again to watch it again, or if you have not been able to watch it. And then I can only invite you to the next webinar. You already had, Nynke already gave you some information on this. So it's on the 14th of June. It will be the last of this series of webinars by Marcus Frederick from BASF in Germany, which indeed, as already mentioned, is about QSAR and read across in the assessment of plant protection products. So I really hope I will see you then during the next webinar. And with this, I wish you a further good day. Bye, everyone.